Uh, can I turn the AC? No? Can I turn the AC? Where's the AC remote? And since it's happening in the USA, there's so many teams from like Africa and like the South, which are literally facing like passport problems because of like Trump's thing. This is their this is the host. Like coaches, players, teams. It's a problem. Oh, beautiful, beautiful. Okay. Let's do this. So we have. Are we done? Yeah. Okay. So we're just gonna finish through mitosis and meiosis. Asexual and sexual reproduction. We're gonna go through both, and after that, then we'll be going into like we're gonna be jumping through a lot because we have notes, and a lot of those notes link to my notes as well. So I don't think I'll read through their notes, but I will like connect it and dot it back, and I'll just kind of see if like I know everything I should know. Apart from that, I think everything's gonna be okay. That's so funny. I do think this is a part of mine will get me mad. That's funny. Okay. So if the camera's not too distracting, the light in the background. It is, but whatever. I like the lens flare. Looks cool. Alright, so already I went through a bunch of this, mainly just the phases of mitosis, and also what's going on, and the phase of interphase as well. G1, S, and then G2. Mostly remember everything. I don't really know. If I were to draw it real quick, so you start off with, so it's P, mat, right? So P is prophase, metaphase, anaphase, telophase, then cytokinesis. And so P is going to be where the chromosomes, well, they should already be like duplicated or like, you know, replicated. So there should be 2 times. So there should be 46 pairs, and essentially they form in the nucleus. They form they condense and become chromatids, and the nucleoid kind of disappears or begins to fade, and also these are called centrioles, but also they are centromeres. Centromeres. Pretty sure centromeres. The centromeres move to the sides or to the polar ends of the cell, and spindle fibers begin to be formed. Then in metaphase, the oh shit, what happens? Anaphase is the lineup. Metaphase is what happens to metaphase? From metaphase they line up, anaphase they begin to be pulled away, and then in telophase then they become what's it called? What's the word called? Like uncondensed. They get released essentially. Anyway, so in metaphase the nucleoid or the nucleus kind of dissipates, and the chromatids line up, and after they line up, the spindle fibers of the centrosomes will bind to the what's it called? To the centromeres of each chromatid. I also remember there's like names like sister chromatid, and then like chromosome. What? Actually, I have no idea. And so then in anaphase, each chromatid is split apart into what? Okay, that's the thing, right? So chromatid is split into um, when chromatid is split, what is the name of the half thing? Is it also still called chromatid? They are not sister chromatids, but as they are called, scientists refer to each single strand piece as an individual chromosome in its own right. Well, technically true. Technically true. It is actually technically one chromosome. Okay, but I'm okay, fine. So each chromosome then moves. So it is then pulled apart to the polar ends of the cells, and the spindle fibers begin to contract. Anything else like this is the cell plate, right? Um, and then in telophase uh, you have the you have the first of all the spindle fibers are like get disintegrated, or they break down, and the chromosomes begin unraveling, so the nucleus starts to reappear, or the nucleoid begins to reappear. Um, and it's kind of like this. I guess like there's like a cell cleavage that forms, that breaks apart in cytokinesis. Well, did we learn about cytokinesis? No, right? Like that's not actually something that we read on yet. So okay. Um, Yeah. Anything else? Not really. That's pretty much it. So okay. Mm, let's see. So I know it's really messy, but it's okay. So let's mark. Okay, so chromosome short. Yeah, long. Okay, longest and which is the shortest? Telophase, right? Or no, anaphase. Anaphase when they get split. So longest phase, chromosomes shorten and thicken to form chromatids. They're held together by centromeres. I don't really remember what terms I use. Centromeres. Then centrioles migrate to the opposite sides. Yes. Centrioles or centrosomes, right? I just call them centrioles. But the thing is, over here, we use the term centrosome, which is the annoying part. Over here, we call them. Oh, never mind. We call them centrioles. Look at that. My fault. My fault, OG. My fault. My fault, OG. So the centrioles move to the polar ends of the cell. Then the nucleus disappears or begins to dissipate. And the end of the phase, you have the nuclear membrane being degenerated, and the spindle fibers begin to form from the centrioles. Metaphase, nuclear membrane breaks down, so it begins to like disappear completely. Chromosome lined up to the equator of the cell, which is the metaphase plate. It's on that cell plate, it's the metaphase plate. Centrioles lie at the poles of the cell. Yes, so they begin to move in prophase, and then they reach the polar ends at the metaphase. Chromosomes are then attached by their centromeres to the spindle fibers. Centromeres is the middle part, right? So the spindle fibers attach at the centromeres, specifically at the kinetic core, right? That's what it binds to. Then in anaphase, the centromeres then split in two, and as a result, if the centromere splits in two, then you know for sure that uh, the thing, the chromosome or the sister chromatids are split apart into singular chromosomes. So then the spindle fiber then contracts, bringing them out towards the opposite poles, and the chromatids follow the spindle fibers. Um, then the final stage of mitosis, where you have the chromatids which have reached, they uncoil and then lengthen. The spindle fibers get broken down. I said the centrioles replicate. I didn't really like that, but the nuclear membrane reforms and the nucleus reappears, right? So all of this, and then also like what do you see at the end? So it says that the each cell will you'll have um, as it undergoes the last stage of mitosis. So this is not talking about cytokinesis. It's actually just specifically talking about telophase. So you'll have two nuclei present in the same cell. You will have um, the, uh, the spindle fibers disappearing. You will have I don't really know, like what would you say? The cell will have signs of cytokinesis. Each cell will have centrioles, last phase, telophase, spindle fibers break down, nuclear membrane. And okay, so they specifically mentioned that the nuclear membrane forms or gets like destroyed, as well as the nucleoli reform and get destroyed. So these are two separate things I need to kind of like pay attention to. Um, Role of the cell cycle here. So we already know that the point of mitosis is to replace, repair, and for growth. And so when we say that the growth of organism is by the cell number increasing, right? So then the asexual reproduction allows the production of clones quicker so that it can grow faster, as well as repair tissues and replace of damaged cells and control the cell growth, all by the same thing. The role of mitosis in the cell cycle in the formation of the morula from the zygote. So this is another thing. Um, Oh, that's a full stop. Each of these are separate points. Okay, shit. Okay. So growth. Can I increase the time? For sure. Don't you want it to them? Yeah, probably. Rubs. So look at each point specifically. So the growth of organism by increasing cell numbers, repair of tissues and replacement of damaged cells, and control of cell growth. So not just the growth of the, because you see, so the cell cycle is also when you have interphase. What's going on in interphase is just the normal metabolic like state, or like it's just doing its normal metabolic like actions, right? It's also like replicating its organelles and its like cytoplasm and stuff. So generally speaking, if you're looking at the basic metabolic like actions that it does during an interphase, when it needs to stop or when it needs to start, that's also being like that is also being controlled by the cell cycle. So the control of cell growth is definitely by the cell cycle. The role of mitosis and the cell cycle in the formation of the morula from the zygote, right? So this is huge, especially when we look at how the zygote, rapidly performs mitosis to become a more developed, creature and survive, right? So that's another thing. Increase in cell number and cells will be genetically identical. I'm not sure how that's a role of the cell cycle. It's like a cause, but I mean that's just an effect. But increase in the cell organelles during interphase. Yep. DNA replication occurs during S phase, so that it can have at the time at cytokinesis, right? So just generally, why do we do DNA replication? So that we can, safely, split the genetic material into two and ensure that the genetic material or the two daughter cells are identical with the parent cell in terms of the genetic information that they hold. Cytokinesis. Now we're learning about cytokinesis. So how does this work, right? So we know that already we have two nuclei. The spindle fiber is already gone. We have kind of like a divide starting to form, but how do we actually, how do we actually break? So a ring of contractile fibers tightens around the cell, which makes a furrow around the outside of the cell, and this cleavage furrow eventually joins up and completely separates the two cells. So I would like to imagine this as the opposite of when a cell fuses, right? Or when a vesicle fuses with the membrane. It like kind of like it kind of, or rather when it breaks off, it like pinches, right? Or is it when it forms? It fuses and then it pinches when it wants to break out. So it's kind of the same thing, right? Where you have the contractile rings around the cell get tighter and tighter and tighter, and so then you have a furrow around the outside of the cell which kind of goes in and out, and that furrow eventually just joins up and pinches and then just separates the two cells. You know, so the contractile ring is over here. It is like where the divide is present. And so the cleavage furrow is obviously this. The contractile rings are here. And so the contractile rings will tighten, making the furrow evident. And then the furrow just joins up, or it just pinches together and separates the two cells. Now, cytokinesis in plant cells is a bit different because now we have, I know there's like a cell plate or something. Plus, there's a plasmodesmata that's also like kind of important, maybe. I'm not completely sure. So, spindle fibers form a barrel-shaped structure in the middle of the cell called the phragmoplast. Various organelles cluster around that phragmoplast. The Golgi apparatus then produces numerous fluid-filled vesicles which fuse to form the cell plate. The cell plate then grows around across the equatorial plane. The vesicles then form the cell walls of the daughter cells, and the cell plate eventually fuses with the parent cell and separate the two daughter cells. So we have that. So we have centrioles, and those centrioles make spindle fibers which form this barrel-shaped structure in the middle of the cell, right? And so we have this barrel-shaped structure in the middle of the cell, and we have various organelles like kind of swimming around it. But most importantly, the Golgi apparatus then produces vesicles. Now, what does the Golgi apparatus do? If you remember, like the pathway of the protein. The protein goes through the rough endoplasmic reticulum, or goes to the rough endoplasmic reticulum's ribosomes to be turned into a polypeptide chain. Then that polypeptide chain is turned into a 3D or 2D structure. When it goes through the rough endoplasmic reticulum. Then when it goes into this smooth endoplasmic reticulum, it is just processed, right? Or does the smooth and the rough do the same thing except the rough just has ribosomes? I don't remember. But the Golgi apparatus modifies the protein, so it adds like a carbohydrate group, so it makes it like glycoprotein, or if it adds like a lipid, then it becomes a lipoprotein, right? Something like that. And then it fill okay, but as a result of the rough or the smooth endoplasmic reticulum, it's going to be sent into or pinched or it's going to be sent out through a vesicle. It was called a transport vesicle, and then the vesicle that the protein is placed in after the uh, Golgi apparatus, Golgi apparatus is going to be the secretory vesicle, which is going to then fuse with the cell membrane and then kind of by exocytosis just remove the enzyme or the protein outside of the cell, right? So um, that's just a recap. So the Golgi apparatus here, we already know if it makes vesicles. So then if it's going to be, so if it's around the fragmoplast, it's going to make vesicles and it's going to fuse to form a cell plate, which grows across the entire plane. So essentially just makes this kind of wall kind of thing, which we call the cell plate. Now eventually the vesicles also form the cell walls of the daughter cells, which the cell plate eventually fuses with the parent cell. So we have the cell plate over here. Then the cell walls also begin to form due to the vesicles, and eventually the cell plate fuses with the parent cell and separates the two daughter cells. It's very kind of weakly explained, but I think I get it. Not really. I should probably like look into it a bit more. Let's see. What I can do is I can could you explain more in depth? What's actually going on in here? Okie dokie. Okie dokie. Okie dokie. Look at beautiful. To understand the plant cytokinesis in depth, we have to look at the mechanical and biochemical challenges the plant faces. Unlike the animal cells, which are flexible and can simply pinch into two using an actin-myosin contractile ring cleavage furrow, plant cells are encased in rigid, unyielding cell walls. So to divide, a plant cell cannot pinch from the outside in. Instead, it must build a brand new cell wall from the inside out. So mitosis transitions from anaphase to telophase. Oh, okay, we're actually talking about PMAT. Okay, so I would imagine P would simply just be the same, where you have the chromosomes condensing, or chromatin condensing into chromat. Oh no, chromat chromatin condensing into chromatids, right? Um, and then what happens is they line up in metaphase and then they get pulled apart in anaphase and then in telophase. Now we're looking at it. So we're seeing in the phase over here that like in the telophase bit, just how they divide specifically in cytokinesis is where they separate, is where they differ. But if when we look at the anaphase to telophase bit, we mark the exact boundaries where the new cell wall must be built, which is the kind of function of the fragmoplast. And so it's made of microtubules, actin filaments, endoplasmic reticulum elements. Um, she said she drew the sarcoplasmic reticulum. Like, what do you mean? Does it just look kind of like the endoplasmic reticulum, which is just like, what does that even look like? It's a single membrane. It's not double membrane. The only double membranes I'm pretty sure is just mitochondria and the other thing, chloroplast. No membrane is ribosome. Um, double membrane is also the nucleus. Okay. I wonder what's like the function of double membrane. Even the animal cells don't have double membranes. So what's up with like mitochondria having it? I guess because you have enzymes inside, like you have the ETC inside the double membrane. No, it's not. It's inside the inner membrane. So why do we have a double membrane? I don't know, sir. Anyway, goddamn, I'm getting a bit tired. Oh, I need coffee, bro. I need coffee so bad. Oh, is the food still like? Is the food over? All the food that you guys bought? Yay. Can I have that for dinner? Can I have that for dinner? Okay. Also, I kind of want to ask Abu, is there a way we could like try planning to move the computer to Hungary, or do you think that'll cause problems with Navi? I know, but just like it's important, man, because our life. Okay, fine. I'll earn and I'll get my own thing. Hmm? What you just said? No, no, no. I just been saying what you said at the farm. I'm broke. Okay, I'm broke. That's the name of the stream. No lie, no lie. Most truthful streamer. Okay. Yeah, Albit's been watching. Albit's been watching. Those who know. Okay. As my like, I'll just say one thing. We were talking on Discord, and I was like, he was sending me some graduation photos. Advit. He was sending me some graduation photos, and and I was like, man, you look so good. Like I look so homeless. Like seriously. And then I was gonna send him a photo, and then he was like, no, bro, you don't. And I'm like, what do you mean? I've seen the streams. He saw me yesterday. It's funny. But I looked pretty good yesterday. Not gonna lie. I was recording my camera. Like while I was studying, I streamed, right. I recorded, so I was recording my camera yesterday on YouTube. Yeah. Sometimes like some friends see it sometimes. Usually it's just Awab, Advit, and some people on Discord. Okay, is your camera on right now? Yeah. Huh? Yeah. You look so bad. Are you watching the stream? Oh, hell no. Okay, shut up. No locking. As mitosis transitions from anaphase to telophase, yep, phragmoplast, blah blah blah. Residual. Okay, how it forms is residual microtubules from mitosis do not completely disappear. It. I'm not watching your stream anymore. I'm not here. I'm not here. I'm sending you a class paper. What is up with the new YouTube? This is not how it usually looks. Oh yeah. That's so. What? Genuinely looks so. I was like, oh wow. Oh, you're like. That used to be. Okay, bruh.
Stop looking at yourself.
Look at this. My reaction. I was reading the, like I was just saying to myself, right? It's like, no, no, it's why are you actually no, 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 no.
Why are you singing?
Shut up,
Go
shut
nowhere.
up, shut up. Shut up.
Hey, what?
My thing is blocked it out of it.
No.
Wait. On the part this way? Hell
Listen, Jack. It's all my fault. No way, man.
I'm thinking weed over here so you can hear me.
That's so embarrassing.
Alright. I've done worse. Way worse. First reading sorry. For so long I've never seen this. Okay, I'm done.
Why did you like it?
You know what's reminds me of this camera? If it was like worse quality, it would look like this camera.
Yeah, I know. Yeah, I know. Silence looks nice and each word is
Okay, okay, down. Down. Zendaya, okay, that's the good side. Uh-huh. Wait,
Where was a bodybuilder?
it's like a six. How do we call it? I really want to come down. Really want to come down. Oh, I really want to come
Oh, I subscribed to it. Is it?
down. Sorry.
It's just, я тут ищу мой
I know.
There's a new meme. It's very good. Sorry, can you leave this one? তাই তো এত ফানি অবস্থায় নাই ফাতিমা খুবই ইম্পর্টেন্ট একটা লাইফের অবস্থানে আছে
Okay. What are we doing? Something about biology. Spindle fibers from mitosis do not completely disappear. Instead, they okay. So after telophase, you know how they're supposed to like disintegrate, right? But they don't. They kind of reorganize and align parallel to each other, perpendicular to the plane of division, which is essentially like this way, I guess. And so the mechanism is that essentially the phragmoplast acts as a physical highway system motor proteins like kinesins, kinesins, move along these microtubule tracks to transport cargo directly towards the center of the cell. Essentially just moving cellulose and proteins towards the center of the cell, I imagine, for the cell wall to be forming. So cargo must be delivered. Cargo in the form of thousands of secretory vessels synthesized with the Golgi apparatus, often referred to as dictosomes. Dictosome. Damn. How do I pronounce this? No, okay. So vesicles packed with polysaccharides. Notice how they say polysaccharides because it is pectin, hemicellulose, and glycoproteins. Okay. Mostly you're containing like some sort of like saccharide, some sort of like carbohydrate in there. Okay. Clustering guided by the phragmoplast tracts, these fluid-filled vesicles crowd into the equatorial plane, creating a dense organized cluster. Then you eventually generate the cell plates, so fusing into networks. So these individual vesicles fuse together to form a continuous hollow tubular network known as the cell plate. Cell plate starts right in the center of the cell as more vesicles are continually guided down the phragmoplast tracts and fuse to the outer edges. The cell plate grows centrifugally. Okay. Duration to the new cell wall. So wait. Oh, okay. So it's not like they release whatever it contains, because like in order to release, they need to fuse into something. So they fuse together and make a network, a tubular network, known as the cell plate. Okay. So eventually it starts at the center of the cell, and as more is added, it's gonna go up and down and fuse to the outer edges, and the cell plate grows centrifugally, expanding outwards towards the existing parent cell wall. So it's going to be moving like upwards over here and downwards over here. Now all of the okay, I think this is not is that actually that's the developing cell plate. That's not the phragmoplast. The phragmoplast is these lines, isn't it? Moving along these phragmoplastic microtubules, right? So yeah. Now, when the cell plate expands and reaches the perimeter of the cell, then its membranes fuse directly with the plasma membrane of the cell, which completes the where is it? Which completes the separation. Membrane fade. The membranes of the Golgi vesicles become the new plasma membrane. The bacterial hemicellulose trapped inside the vesicles undergo a biochemical cross-linking to mature into the middle lamella, as well as I imagine the primary and the secondary cell walls as well. Yeah, the deposition of cellulose is usually what it means. So pectin hemicellulose trapped inside the thingy makes the middle lamella, and that's the sticky cementing layer that glues adjacent plant cell walls together. So Deposition of cellulose. Once this boundary is sealed, the daughter cells activate enzyme complexes, cellulose synthase on their new plasma cell membranes to lay down rigid cellulose microfibrils completing the primary cell wall. Okay. Yes, understood. Given that the phragmoplast must expand towards the cell cortex to guide these vesicles, how do you think the cell ensures that the phragmoplast expands precisely towards the correct predetermined cell? I have no idea. Like, I could not tell you. This is just like one of those things I just like, you just ignore. It just does that. Whatever. It just goes there. It just knows to do that. Which is like my experience with a lot of this subject. Anyway, that's mitosis done for both plant and animal cells. Whew. As meiosis is gonna be it's gonna take a while, not too long, hopefully. I don't want to read all this. I don't. I really don't. But I should. I did not study any of those. What? I do remember this though. Whoa. Wait, that's not good. Mm, 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 mm. Oh my god, there's so many. Normal distribution. I mean, okay, gene expression isn't that hard. Or at least I didn't find that difficult. Anyways, I don't think I'm planning to do all of this. I think we should just stick with let's not go into environmental influence on genes just yet, even though they do talk about genotype and phenotypes, which is important. But I actually think that I should know what's before this to get to this spot, right? Because I don't even know what I don't really like my background is not good. So yeah. I also like to look at like the other stuff, like what was it called, Mendeleev's stuff, which I maybe was in unit 1. I don't actually quite remember. Actually, I think we should be able to open it. Maybe it's here. So, oh my god. Oh my god. Okay. Do. These notes look so weird and simple. They kind of look like my school notes. Interesting. Maybe near the end? Yeah, there you go. So genetics, what was before this? Mendelssohn installs experiment. Okay. Oh, DNA replication. Okay, wait, this is huge. I would not mind reading all of this. Oh, and also, wait, what is this? Oh my god. Dude, do you remember that table? And then we had to learn the different kinds of mutations. Man. Damn. 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 Hmm. So we're learning about the central dogma. Ah, it's too big. It's too big. Ah, here we go. It's the table. Hmm. All of this I'm really bad at. And I'm pretty sure they talk about the different shapes of proteins right above this as well, which I'm also pretty bad at. Hmm. This is fine. Oof. Okay. This I keep on messing up. I actually do keep on messing this up. Oh yeah, and the effects of like increasing temperature and stuff. Okay. Whoa. Huh. I ignored all of this. Hmm. Okay. It's a lot of information. A lot. But we'll take it one at a time. For now, let me just bring the stuff back so just we kind of have an idea of what we're working with. So okay. So my meiosis. So one stage in gamete production. Um, so we have one stage where we form the duplicates. Okay. Production division occurs in the blah 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 blah. It is the basis of variation that allows the species to evolve. Two nuclear divisions occur and give rise to four haploid daughter cells. Each daughter cell has its own unique combination of genetic material. So, okay. Basis of the variation that allows the species to evolve. Two nuclear divisions occur and give rise to four haploids. So, multiple roles or multiple features that we need to like kind of pay attention. It it takes place in like or it has something to do with gamete production, and obviously we need gametes for fertilization, for literally just like reproduction. Um, the reduction division and occurs in these kind of organs. So where is over here. And it is the basis of the variation that allows the species to evolve. So this is like evolving, like kind of this is that in that sense. Two nuclear divisions give rise to four haploid daughter cells rather than two identical diploid cells. We have four unidentical haploid cells or similar haploid cells. Um, each daughter cell has its own unique combination of genetic material. So this is unique to the parent. Okay. Now, oh god, I'm so tired. There. Damn. Alright. Wow. Speaking gets me so tired. I'm trying to play the cover. आगे दिखते पारबा দিলে আমি নাও তাড়াতাড়ি করে নামাজ পড়ে আমি ঘুমাতে পারবো, সো আমি ফজর জন্য উঠতে পারি। I've been on a roll with Fajr, alhamdulillah. Today, yesterday, day before, I've just been waking up like right during Fajr time. Blessed. Hashtag blessed. Alright, so homologous pairs of chromosomes. Chromosomes replicate before division, so this is not meiosis. I mean, this step is not meiosis when it replicates. This is, I mean, you can imagine this is going through the S phase and as well as the G phases to get everything ready, the cytoplasm, the other organelles. After replication, each chromosome is made out of 2 strands of genetic material called the 2 chromatids. There is no need for, we don't talk about condensing, it's already condensed. So all we need to think about is in male, so all we need to think about is in the pre-meiosis, we have the condensed chromatids already. We have the replicated chromosomes already. We have the replicated chromosomes already, the condensed chromatids already, and yep, that's pretty much it. And so in meiosis 1, the homologous chromosomes pair up and then separate. So they pair up in the center of the cell, the homologous pairs. So over here, like let's say this is a homologous pair of chromosomes, right? So when it's replicated, when each of these replicates, so then we'll form something like this. So now we have homologous pairs of replicated chromosomes, okay? Or chromatids, if you want to call them. So then now, when these chromatids line up and pair up together, then we kind of separate. You could imagine that it's probably like pulling apart just like anaphase, like just like whole PMAT cycle as well as cytokinesis. And then what happens over here is that there is another set of the cytokines here, which bind in the same exact way and replicate once more to leave us with only no longer a pair of homologous, but one single homologous chromosome. Or it's not really chromosome here; it's haploid, but one single homologous chromosome in 4 of these. Okay. Now, one important thing is that the query mentioned where the variation arises from, but I'm trying to remember because I remember studying this like so in depth, like crazy in depth, and I'm pretty sure I have it recorded on the channel as well, which is insane. Anyways, so the chromatids separate and the gametes are formed with each with half of the original number of chromosomes. Okay, okay here we have the actual phases. So in prophase 1, okay, they do condense up into chromatids and homologous pairs of chromosomes associate with each other. What do we mean by homologous pairs of chromosomes associate with each other? This is something I'm kind of confused with. Like, what is a homologous? Like, what makes them homologous? So I'm a bit confused here. What do they mean by homologous? How are they homologous? Okay, so homologous fundamentally means having the same structural features and patterns of genes. Understand why these chromosomes are pairing up in prophase 1, we must, okay. Having same structural features and patterns. Origin and genetic architecture. So, in maternal and paternal, in a diploid organism, diploid meaning it has 23 pairs, you in this organism you would inherit two complete sets of chromosomes. One set from your mother and another set from your father. And each homologous pair consists of one maternal chromosome and one paternal chromosome that must match up with each other. In the image you provided, the red, notice how the top pair has a red and the matching is a green, and the bottom one is a green matching with a red. So those two are distinct homologous pairs. What makes them match is not that they are identical, but they carry the same genes at the exact same location. Oh, I remember this. You have the loci. Dang. Yeah, I remember that. It's right here somewhere. Oh my god. No. Right here? Yeah. Nah, I should have studied this. Oof, dude. Yeah. Anyway, so it's like whether or not it's chromosome 1, 2, 3, all the way to 23 depends on like the genes in the what the genes code for in the loci, right? Homologous chromosomes are morphologically identical. Oh, under a microscope, a homologous pair will display the exact same total length, centromere positioning, whether if it's in exactly the middle or shifted towards an end, staining band patterns. So during mitosis, the homologous chromosomes completely ignore each other. However, in prophase 1, they actively try to find each other and then physically lock together in a process called synapsis. This precise alignment is critical because it allows them to undergo crossing over, where they swap and break corresponding segments of the DNA, which shuffles the maternal and paternal, allows creating genetic variation, which is essential for biodiversity. So, God. Can you please? I'm really tired from doing nothing all day. Hmm? I do money. Bro. Okay, I'm gonna take like a 5 minute nap. Do, 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 do.